Have you ever wondered what's going on inside that giant oak tree or that delicate little wildflower?
It's amazing, isn't it, how these incredible organisms just thrive seemingly effortlessly.
Well, today on the deep dive, we're going to try to crack the code. We're diving deep, no pun intended, into the fascinating world of plant anatomy, specifically the anatomy of flowering plants.
Yes, using some really detailed studies as our guide. We're going to try to understand how their structure, their internal framework, lets them survive and thrive in pretty much every environment on Earth.
So think of this deep dive as a mission, a quest to understand plant design, their blueprints, if you will. Because once we grasp how it all works inside, we can really start to understand the incredible adaptations that make plants so successful.
It's going to be a journey through layers, starting with tissues, then organs, and then seeing how it all comes together.
And we'll focus on the three main tissue systems that make up plants: the epidermal, the ground, and the vascular. These are the fundamental building blocks.
And what's really cool is that even within the flowering plants, there's so much variation, like take monocots and dicots.
Right. Different internal arrangements, different strategies for life, and these structures can even change depending on the environment the plant is in.
Talk about adaptation. It's incredible how plants can modify their structure to suit their surroundings.
So let's start peeling back these layers, shall we? First up is the epidermal tissue system. I like to think of it as the plant's skin.
Perfect analogy. It's the outermost layer, the protective barrier between the plant and the outside world.
And it has to do a lot, right? Not just protection, but also regulating what goes in and out.
Absolutely. It's a busy layer, for sure. And there are a few key players in this epidermal defense force, so to speak.
All right, let's meet them. Who are the main characters?
Well, first you've got the epidermal cells themselves. They're kind of like the bricks in a wall, elongated and packed tightly together to form that continuous layer. Usually it's just a single layer of these cells.
So a thin but strong shield.
Yeah, you could say that. Now, these cells are what we call parenchymatous, which means they have thin cell walls.
Thin walls, but they still need to hold things together.
Right, and that's where the vacuole comes in. Each epidermal cell has a large central vacuole.
Ah, the vacuole, the storage tank of the cell.
More than just storage. Think of it like a water balloon inside each cell, helping maintain pressure and keep the plant firm. It's called turgor pressure.
So it's all about balance, right? Strong, tight cells for protection, but with that inner pressure to maintain rigidity.
Exactly. Now, we can't forget about the openings in this wall, the stomata.
Right, the stomata. These are crucial for gas exchange, like breathing in plants.
Precisely. They allow carbon dioxide in for photosynthesis and let out oxygen. And importantly, they regulate water loss through transpiration. Stomata regulate gas exchange and transpiration. Stomata regulate gas exchange and transpiration.
So they control what comes in and what goes out.
They're like tiny gatekeepers. Each stoma is made up of two specialized cells called guard cells.
Guard cells, like little bouncers at the club.
Kind of. They're usually bean-shaped, and they enclose the actual opening, the stomatal pore.
And in grasses, they're dumbbell-shaped.
Yes, good catch. Now, these guard cells have a special trick up their sleeve. Their inner walls, the ones facing the pore, are thicker than their outer walls, and this difference is what allows them to open and close, controlling the flow of gases and water vapor.
So it's like a tiny valve system responding to the plant's needs.
You got it. And these guard cells, they even have chloroplasts, just like the cells in the leaves that carry out photosynthesis.
But wait, aren't stomata mainly on leaves? What are chloroplasts doing there?
Well, the chloroplasts in guard cells don't seem to be directly involved in making food like in the leaf cells. Instead, they seem to play a role in regulating the opening and closing of the stomata.
Fascinating. And sometimes there are other cells surrounding the guard cells, right?
Yes, those are called subsidiary cells. Together, the pore, the guard cells, and any subsidiary cells make up what we call the stomatal apparatus.
The whole shebang, the entire team working together.
Exactly. It's a well-coordinated system for regulating the plant's internal environment. Now, aside from the cells and pores, the epidermis can also have these interesting outgrowths, like hairs.
Hairs. On plants.
Oh, yeah. They're called epidermal appendages. You've got trichomes on the stems and root hairs on the roots. Let's start with the root hairs.
Right. Root hairs first. What do they look like?
Imagine tiny little fingers extending from the epidermal cells of the root. They're actually just single-celled extensions, but they make a big difference.
I'm guessing it has something to do with their job, absorbing water and minerals?
You're sure. That's exactly it. They increase the surface area of the root, maximizing contact with the soil. More contact means more absorption. Root hairs absorb water and minerals. Root hairs absorb water and minerals.
So it's like having a giant sponge underground, soaking up all the good stuff.
A very efficient sponge. Now let's move up to the stem and talk about trichomes.
Okay, trichomes.
Yeah.
What's their role?
Well, trichomes are usually multicellular, and they can be all sorts of shapes and sizes. Some are branched, some are unbranched, some are soft and hair-like, others are stiff and prickly.
So a variety of defenses, depending on the plant and its environment.
Exactly. And some trichomes even produce special substances like oils or resins. But one of their main jobs is to help prevent water loss from the stem.
Ah, so they act like a little fuzzy coat trapping moisture?
Precisely. They help create a layer of humid air close to the plant's surface, slowing down evaporation. Trichomes help prevent water loss. Trichomes help prevent water loss.
Clever. So both root hairs and trichomes are about maximizing surface area, but for different purposes.
Right.
Absorption below ground and water conservation above ground.
Spot on. And we can't forget about the cuticle. This is a waxy layer covering the epidermis of most stems and leaves.
The cuticle, that sounds familiar.
It's like nature's sealant. Its main job is to prevent water loss. The cuticle prevents water loss. The cuticle prevents water loss. Think of it as a waterproof jacket for the plant.
Essential for survival, especially in dry environments.
Absolutely. Interestingly, you don't find the cuticle on roots. Their job is to absorb water, not prevent its loss.
Makes sense.
Yeah.
Okay, we've spent a lot of time on the outermost layer. What's next?
Let's dig a little deeper, shall we? Underneath the epidermis lies the ground tissue system.
The ground tissue system, what's its main role?
This is where you find the bulk of the plant body. It's everything that's not epidermis or vascular tissue.
So the filling in the sandwich.
You could say that. It's involved in storage, metabolism, and providing support.
Busy system.
Definitely. And it's made up of 3 simple tissue types: parenchyma, collenchyma, and sclerenchyma.
Okay, let's break those down one by one. What's parenchyma like?
Parenchyma cells are the most common type in plants. They're thin-walled and usually have spaces between them, allowing for gas exchange and storage.
So they're kind of the jack-of-all-trades cells.
Yeah, pretty much. You'll find them in the cortex, which is the region between the epidermis and the vascular tissue. Also in the pericycle, just inside the endodermis, and in the pith, the central core of the stem and root. And then there are the medullary rays, which are like bridges connecting the pith to the cortex. Parenchyma is found in the cortex, pericycle, and pith. Parenchyma is found in the cortex, pericycle, and pith.
Wow, they get around. And what about leaves? Do they have parenchyma too?
Absolutely. In leaves, the parenchyma tissue is called mesophyll, and it's packed with chloroplasts.
So this is where the magic of photosynthesis happens.
Exactly. Mesophyll is the primary site of photosynthesis in the plant. Mesophyll carries out photosynthesis. Mesophyll carries out photosynthesis.
Okay, on to collenchyma. What's its specialty?
Collenchyma is all about flexible support. It's especially important in young, growing parts of the plant, like stems and leaf stalks. Their cell walls are thickened unevenly, which gives them strength, but also lets them bend.
So they provide structure, but allow for growth.
That's the key. Think of the strings in celery. That's collenchyma.
I always wondered what those were. And finally, sclerenchyma. What's its role?
Sclerenchyma is the heavy lifter. It provides rigid, strong support to the plant. They have really thick cell walls and are often dead at maturity.
Dead. Like completely dead.
Yeah, their job is structural, so they don't need all the living cell machinery anymore. They basically become little bricks holding the plant up.
Wow. Nature is so efficient. And there are different types of sclerenchyma, right?
You're right. There are sclerides, which are short and often give fruits like pears their gritty texture. And then there are fibers, which are long and slender, like the ones used to make linen or rope.
So we've got the parenchyma for storage and metabolism, collenchyma for flexible support, and sclerenchyma for that rigid strength.
Perfect summary. And all of these working together make up the ground tissue system.
Amazing. Now let's talk about the plant's highway system, the vascular tissue.
Yes, the vascular tissue system. It's like the plant's plumbing, transporting water, minerals, and sugars throughout the entire plant.
So there's a constant flow of materials going on inside.
Absolutely. And it's all thanks to the xylem and phloem. The xylem carries water and dissolved minerals upward from the roots, while the phloem transports sugars from the leaves, where they're made during photosynthesis, to other parts of the plant.
So a two-way system, keeping everything connected.
Yes, a vital network. Now, xylem and phloem are usually found together in bundles called vascular bundles.
Vascular bundles, and these bundles are arranged differently in different parts of the plant, right?
Exactly. There are 2 main types of arrangements: radial and conjoint. In radial vascular bundles, the xylem and phloem alternate like spokes on a wheel. This is characteristic of roots. Radial vascular bundles are in roots. Radial vascular bundles are in roots.
Okay, radial and roots. What about stems and leaves?
In stems and leaves, the vascular bundles are conjoint. This means the xylem and phloem are together on the same radius, with the phloem usually on the outer side of the xylem. Conjoint vascular bundles are in stems and leaves. Conjoint vascular bundles are in stems and leaves.
So two different strategies for organizing the transport system.
And within these conjoint bundles, there's another distinction: open versus closed.
Open and closed. What's the difference?
It all comes down to cambium, a special kind of tissue that can produce new xylem and phloem cells, allowing the plant to grow wider. Open bundles have cambium, while closed bundles don't.
So open bundles mean the plant can grow thicker over time.
Precisely. Open vascular bundles allow secondary growth in dicot stems. Open vascular bundles allow secondary growth in dicot stems. This is why dicot stems can become woody, like in trees.
And closed bundles are found in monocots, which don't typically get woody.
That's right. Closed vascular bundles do not allow secondary growth in monocots. Closed vascular bundles do not allow secondary growth in monocots. They stay relatively slender.
Okay, this is starting to paint a clear picture of some key differences between monocots and dicots.
Yeah, these differences are fundamental. Let's delve a bit deeper into their root structure to see these variations.
Roots, the hidden heroes of the plant world.
Yeah.
What are their defining features?
Both dicot and monocot roots have an outer layer called the epidermis, sometimes called epiblema, which has those root hairs we talked about.
Right, the tiny fingers grabbing water and nutrients.
Exactly.
Yeah.
Then there's the cortex, made of parenchyma cells with spaces for air, and the endodermis, which acts like a filter, controlling what enters the vascular tissue.
So the basic layout is similar.
Yeah, the basic blueprint is there, but there are key differences. For example, the number of xylem bundles. Dicot roots usually have 2 to 4. Dicot roots have 2-4 xylem bundles. Dicot roots have 2-4 xylem bundles. Monocot roots, on the other hand, have many more. We call it polyarch.
Okay, so more xylem bundles and monocots. What else?
The pith, that central region. It's small or even absent in dicot roots, but it's large and well-developed in monocots. Monocot roots have many xylem bundles and a large pith. Monocot roots have many xylem bundles and a large pith. And remember, dicot roots can have secondary growth, while monocot roots generally don't.
So dicot roots have the potential to get thicker, while monocot roots stay relatively slender.
Exactly. Now let's shift our focus upwards to the stems.
Okay, time for a stem comparison. What are the highlights?
Dicot stems have an epidermis with a cuticle, sometimes trichomes, and a few stomata. Then comes the cortex with a collenchyma layer for support, followed by a parenchyma, and then the endodermis, sometimes called the starch sheath.
A lot going on in that cortex.
Definitely. Then there's a pericycle with sclerenchyma patches, and finally, the vascular bundles arranged in a ring around a central pith. Those pith rays connect everything together. Dicot stems have vascular bundles in a ring. Dicot stems have vascular bundles in a ring. And each bundle is open, meaning it has cambium for secondary growth.
So that ring of vascular bundles is a dead giveaway for a dicot stem.
Pretty much. Now, monocot stems are quite different. They also have an epidermis and a sclerenchyma layer for support, but their vascular bundles are scattered throughout the stem, not in a ring. Monocot stems have scattered vascular bundles. Monocot stems have scattered vascular bundles.
So scattered bundles instead of a ring.
Exactly. And these bundles are closed, no cambium, so no secondary growth. They also lack phloem parenchyma, but might have water-containing cavities instead.
So many fascinating variations. Finally, let's talk about the food factories. The leaves.
Ah, the leaves. Dicot leaves are usually what we call dorsiventral, meaning they have distinct upper and lower surfaces. Both surfaces have a cuticle, but more stomata on the bottom.
Makes sense to have more stomata on the underside, protected from direct sunlight.
Exactly. Now, the mesophyll, that's the photosynthetic tissue, is divided into 2 layers. Palisade parenchyma on top, with tightly packed cells full of chloroplasts, and spongy parenchyma below, with more loosely arranged cells and air spaces. Dicot leaves have distinct palisade and spongy mesophyll.
Dicot leaves have distinct palisade and spongy mesophyll.
So a division of labor, optimizing light capture and gas exchange?
Exactly. And then there are the veins, containing vascular bundles of different sizes, forming a network pattern called reticulate venation.
Dicot leaves are like little solar panels with a branching network of pipelines. Great analogy. Now, monocot leaves are different. They're often isobilateral, meaning the upper and lower surfaces are similar, and they have stomata on both sides. Monocot leaves have stomata on both sides. Monocot leaves have stomata on both sides.
Interesting. And the mesophyll?
It's not differentiated into palisade and spongy layers. It's just a uniform layer of parenchyma with chloroplasts. Monocot leaves have undifferentiated mesophyll. Monocot leaves have undifferentiated mesophyll.
So simpler structure in monocot leaves.
You could say that. Their vascular bundles are also similar in size, reflecting their parallel venation pattern.
Parallel veins, like
Yeah.
train tracks running along the leaf.
Exactly. And some monocots, like grasses, have special bulliform cells that help the leaves curl up during dry periods to conserve water.
Wow, so many clever adaptations. This has been a whirlwind tour of plant anatomy, and we've covered so much data about these amazing organisms.
It's incredible, isn't it? From the epidermal tissue system, that protective outer layer, to the ground tissue system, responsible for storage, metabolism, and support, to the vascular tissue system, the plant's internal transport network, every part is perfectly designed for its function.
And the key takeaways, those data points that really stick with you, are the differences between monocots and dicots. Whether it's the arrangement of vascular bundles, the presence or absence of cambium, or the structure of the mesophyll, these details reveal so much about how these plants function and thrive in different environments.
Absolutely. Understanding plant anatomy is like having a key to unlock the secrets of the plant world. It's essential for anyone interested in agriculture, horticulture, botany, or even just appreciating the plants around us.
So the next time you see a plant, whether it's a towering tree or a tiny blade of grass, remember the incredible complexity hidden within. Think about the intricate tissues, the vascular bundles transporting water and nutrients, the chloroplasts capturing sunlight, and the countless adaptations that have allowed plants to conquer nearly every corner of our planet.
And keep in mind, there's still so much more to discover. The world of plant anatomy is full of wonder, waiting to be explored.
Thanks for joining us on this deep dive into the fascinating world of plant anatomy. We hope you've gained a new appreciation for the intricate data that makes plant life possible.
It's been a pleasure sharing these insights with you.